we're talking about DNA structure again, but it's just going to be a brief overview. I'm going to see what you remember, and then you have an activity that I'll explain later. So we're going to start off by doing some warm-up questions. I'm going to hide your name, so don't worry. No one's going to see who you are. I think we did Himalaya last time, so I'll do space this time. So I'll go ahead and send the link again in the chat room. We're doing our notes on Nearpod today. I think I'm just waiting on a couple of more people. All right, I'll go ahead and start anyways. DNA is found in the blank of eukaryotic cells. Eukaryotic cells. That would be the nucleus. They're found in the nucleus. Next question, DNA is found in the blank of prokaryotic cells. Prokaryotic cells. That should be the cytoplasm. What are the three main functions of DNA? Three main functions of DNA. I think that's storing, copying, and transferring, which is not, oh, sorry. What are the building blocks of nucleic acids? Building blocks of nucleic acids. That would be the nucleotides. Next question. What part of the DNA molecule is also known as the sugar molecule? That's the deoxyribose. Y'all are doing great. What does thymine always bind with? Thymine. That is with adenine. Next question. What does guanine always bind with? Good job, guys. Yes, that's cytosine. Next question. What is the name of the twisted ladder model of DNA? And that is the double helix. So good job to my top 3 people, my giraffe and my 2 penguins, and good job to everybody else who played. If you don't remember this stuff, go back to yesterday's notes and review again. All right, so deoxyribonucleic acid is a molecule that carries genetic information in all living things. We have two nucleotide chains that connect together. They're going to connect like a ladder, and that ladder is going to twist around each other like a spiral staircase, and that's where we get the name double helix from. So the DNA is structured like a double helix, a twisted ladder. Its three main functions include storing information like your eye and hair color, copying information so that way we can make more cells, and transmitting information so we can pass on our genes to our other cells. Work like that. DNA is a nucleic acid made up of nucleotides. Each nucleotide is made up of three important compounds: a deoxyribose 5-carbon sugar, a phosphate group, and a nitrogen base. Remember, there's a saying to help you remember the correct order. Pancakes smell nice. Pancakes is for the phosphate group. Smell is for the sugar. And nice is for the nitrogen base. Nucleotides are joined together by covalent bonds that are very, very strong because we don't want our nucleotides to break apart. We need them to stay together. The sides and the backbones are going to be the phosphate and the sugar. And that's what makes up the sides of our DNA molecules. All right, so go ahead and label. Oh, I'm just having you color. You're coloring. Color the nucleotide. Color the parts of the nucleotide. Color the nitrogen base, the deoxyribose, the phosphate group, and then color the covalent bonds. Got some beautiful coloring going on. All right, so the majority of you got this down. Yes, the red nitrogen base is the square. The deoxyribose sugar is the pentagon. The phosphate is the circle. And then the lines would be the covalent bonds, which technically they just connect more nucleotides together, but I couldn't find an image like that. So we're just going to pretend those are the covalent bonds. So a nitrogen base works as the genetic code for the cell. Basically, it gives the instructions for your cell. The way they're ordered and arranged is how our cell knows what to do. It also explains what we're going to look like based on their order and arrangement. So DNA has 4 different kinds of nitrogen bases: adenine, thymine, guanine, and cytosine. Those are the 4 kinds of nitrogen bases for DNA. We're going to learn later on that RNA has something different, but we're not going to focus on that yet. I just want you to pay attention to these main 4. These nitrogen bases are going to be connected to each other in the center of the ladder with hydrogen bonds. And those hydrogen bonds are really weak. And they're weak because we need to break them apart so that way we can make more copies of our DNA. So again, they're really, really weak. There is a saying to help you remember how the nitrogen bases pair up, because they're always going to pair with the same nitrogen base no matter what. We can't mix them around. It doesn't work like that. Adenine and thymine are always together. AT, always together. They're always going to be paired with each other. Anytime you see an A, it's going to go with a T. Anytime you see a T, it's going to go with an A. And then guanine and cytosine stand for grandma's cookies. Again, G and C are always going to go together. You will always see them together. Just keep in mind that DNA is also anti-parallel. I talked about that yesterday. They don't connect with similar structures. The right side's always going to be flipped, so that way it could connect easier. So just keep that in mind when we're connecting them later today in today's activity, but I'll show you how to do that. All right, you're going to select the matching pairs with their complementary DNA strand. Basically, what is it going to pair up with? There are wrong answers. Good job, guys. All right, so let's explain how they match, because I see that we got a little confused there. So they're always going to be opposite of each other based on what they connect to. So for example, right here on the top left, I have CGT. Now I need to find what it's going to connect to. Well, that C is only going to pair with the G. The G is only going to pair with the C. And that T is only going to pair with an A. So CGT will pair with GCA. On the next one, I have AAG. The only one that A is going to connect with is T. So I have TT. And then my G is only going to connect with the C. So my letters AAG will pair up with TTC. And that's how they're going to pair together. All right, let's try that one more time. Go ahead and match them together. Whoops, I don't want to show your names. There we go. All right, well, we did a lot better on this round. If this is still confusing you, make sure you stay on after Zoom so that way I can show you how to do it, because this is part of today's assignment and I don't want you to get a bad grade on it just for not being able to match correctly. So I'm going to explain it one more time how to match them. We have a TCT right here. T can only connect with A, C can only connect with G, and T can only connect with A. So TCT can only connect with AGA. Remember, AT always together, GC, grandma's cookies. All right, so DNA is specific for you. No one in the world will have the same DNA as you unless you have an identical twin. So yes, identical twins do have the same DNA structure. Their fingerprints are different, but they'll have the same DNA. So it's kind of really hard to tell a paternity test or a maternity test with identical twins. Fraternal twins, different, but identical twins have the same DNA. All right, so question. Will the DNA in your eye cells be the same as the DNA in your stomach cells? Think about that. Will your eye cells have the same DNA as your stomach cells? Think about that one for a minute. All right, hopefully you got your answer in your head. So yes, they are going to have the exact same DNA. Your cells are all identical in your body. Every single cell in your body is going to have the same DNA code as every other cell in your body. They're all going to be the same. So that means your eye cell has the same exact DNA as your stomach cell. It has the same exact DNA as your toe cell, as your hair cell, as your urine samples. Now the next question is, do you think this is too much knowledge for a cell? How does the cell balance it all? So think about that. If our cells have all the DNA, is that too much? How does the cell balance it all? So your cells are going to balance all of that information with something called gene regulation. The cells in your body don't activate every gene at every single time. Your eye cell will only activate the information needed for its location. The same goes with the stomach cells, and the other genes will be inactive. So in your eye, the cells in there, they're only going to turn on the DNA that's needed for that location, for that eye. Uh-oh. Okay. It's going to turn off all the other DNA instructions for your stomach, for your toes, for your intestines. It's going to turn all of that off, and it's only going to turn on the stuff it needs for that eye. Gene regulation is the process by which cells determine which genes will be active and which genes will not be active. It also makes the cell decide to become a red blood cell, a neuron, or any type of cell in the body. That's what gene regulation does. It's going to tell the cell what to be, and it's going to tell the cell what to do, depending on where it's at in the body. So it's going to turn off the other information needed. It's kind of like when people say you only use like 10% of your brain. It's not saying that all 90% is just dead. It's just saying you only use 10% at a time. So 10% here, then 10% here, then 10% here. That's what that means, and that's what gene regulation does. All right, so this is a video about why your DNA is unique to you. I'm gonna play it. I don't have questions. It's just a video.
What is DNA fingerprinting or DNA profiling? Leicester University geneticist Alec Jeffreys developed a technique called DNA fingerprinting in 1985. It allows DNA samples from different people to be compared to look for similarities and differences. It's used for solving crimes and can also confirm if people are related to each other, like in paternity testing. Any two people in the world have 99.9% of their DNA the same. So this process analyzes the differences in the remaining 0.1%. This modern technology is called DNA profiling. It's a very sensitive technique which only needs a few skin cells, a hair root, or a tiny amount of blood or saliva. There are sections, or loci, of chromosomes, where instead of a gene consisting of a long sequence of bases, there are much shorter sequences of 3, 4, or 5 bases that are repeated many times. For example, these repeated sequences are called short tandem repeats, or STR. At these places on the chromosomes where we find these STRs, there are areas that vary in number of repeats. DNA profiling only looks at these STRs. A cell sample is collected. This could be from some blood at a crime scene, or a swab from the inside of someone's cheek, for example. The DNA is then extracted from the sample. Many copies of this DNA may be made using the polymerase chain reaction, or PCR. Special enzymes called restriction endonucleases are used to cut the DNA up into different size pieces. The DNA samples are then put into wells in a special gel called agarose for the process of gel electrophoresis, which separates the DNA fragments by size. The pattern is then transferred to a nylon sheet in southern blotting. And finally, the lines produced by the DNA samples from different people are compared. So let's have a go at using DNA profiles to solve some mysteries. An item was stolen in a burglary. A drop of blood was left behind by the thief. Samples of DNA were taken from 4 suspects and compared to the sample left at the crime scene. Which suspect is guilty? Here's a clue. Look for the one that is most similar to the one from the crime scene. Pause the video while you decide.
All right, so take a look at the blood samples. Sorry, there's a giant play button in the middle. But take a look at it and take a guess at which one is the criminal. Do you have a guess in your head? All right, so I think they explained it in the video.
Suspect 3 is guilty. Can you see how the pattern of bands matches in suspect 3 and the crime scene?
So that's actually how crime investigations work. When we have blood samples, when you have urine samples, saliva samples, this is actually how they prepare that blood work based on the DNA to compare it to see if they're the ones that were at the crime
Their
scene.
DNA is the same. Let's try another. A soldier has been killed in an explosion and has lost his dog tags that identify him. 3 soldiers are missing from their unit, so the army asks the 3 sets of parents for a DNA sample so that they might compare it to the soldier's DNA and therefore make an identification. Remember, the soldier will only share half of his DNA with each parent. So which set of parents is the soldier the son of? Pause the video while you decide.
So when you're doing paternity and maternity tests, a child can't have a DNA molecule, a line that doesn't come from a parent. So for example, the soldier has a very thick line at the top, but parents A and B don't have that line at all. He had to have gotten it from somewhere. So since these parents don't have it, it can't be parents A and B. Parent C doesn't have it, but parent D does. Well, he could get it from D. He doesn't necessarily have to get it from C. Parent D could have given it to him. And that's how that works.
Parent C and D. As you can see, he inherited the first band from parent D and the second, third and fourth from parent C, and so on. So now you understand how DNA profiles or fingerprints are made and seen some examples of where they can be used. You can even interpret a DNA profile.
All right, so now we're going to label a DNA strand. tell me which ones are the adenines, which ones should be the thymines, which ones should be guanine and cytosine. And then again, color your phosphates, your deoxyribose. Color your covalent bonds dark brown and your hydrogen bonds light blue. So good job. Yes, all your circles should be blue. All your A's should be red. All your T's should be orange. All your guanine should be green. All your cytosine should be yellow. All right, so I'm going to use this one as an example because they finished and they did everything correctly. The one thing I want to point out, everything else is correct, but the one thing I want to point out that's most important is that the covalent bonds right here are all on the outside. Those are the dark brown lines. Your hydrogen bonds are the light blue ones, and those are the ones all on the inside. So that's correct. Everything on this is correct. Good job. All right, so now we're gonna do the DNA alias model activity. I promise you it's not that hard. The hardest part is probably matching them up correctly. So we're gonna type your initials in the box below. If you have more or less than 3 initials, you can just use the first 3 letters of your name. My initials are VAT. Those are my initials. Now I'm going to use the table to help convert my name into its DNA alias. I'm going to look each letter of my name, and I'm going to look at the codon column to see what it's going to make. So my first letter is V. V makes GTC. So I'm going to type GTC. Then I'm going to add a dash. My next letter is A. A is GCT. So I'm going to type GCT, then I'm going to add a dash. And my last letter is T. That's ACT. So I'm going to add ACT. So now I'm going to make my DNA strand using my letters. I have GTC. So G, T, C. And then I have GCT. So that's GCT. Then I have ACT. So that's ACT. So I'm going to find their matching pairs. G goes with C. T goes with A. And C goes with G. So if you're reading the instructions, it says that you have to turn these around because You know, they're mirrored images of each other. So this one has to be flipped like that. Remember, we want the phosphate and the sugar on the outside. So you're just gonna have to flip everything on your left side upside down. Shouldn't take you long. So obviously I'm gonna know if you cheat because it's based on your initials. Don't use somebody else's initials. That's pretty much it. Easy peasy, you got this. If you need any help or if you have any questions, stay on. If not, you are free to go.